Hello and welcome to Lab 2. Today we will discuss how to do the interfacial tension measurement. The applied setup is called pendant drop tensiometer, which can also be used to measure the surface tension and contact angle. Interfacial tension is a measure of how much energy is required to make a unit area of interface between two immiscible liquids. It is a concept of fundamental importance in colloid science, describing phenomena as diverse as the formation, shape and stability of liquid drops, the surface energy cost in forming an emulsion and the force applied by a capillary liquid bridge. Determination of the interfacial tension allows deductions to be made regarding the chemical composition of fluid interfaces and the adsorption and desorption of surface active solutes. The analysis of the drop shape is based on the Young-Laplace equation. This equation describes the pressure difference Laplace pressure between the areas inside and outside of a curved liquid surface, interface with the principal radii of curvature. The forces that determine the shape of the pendant drop are in particular the surface tension and gravitation. When at the vertex, R1 equals to R2. The surface tension seeks to minimize the surface area and get the drop into a spherical shape. Gravitation on the other hand stretches the drop from this spherical shape and the typical pear-like shape results. The introduction of a parametrization using the arc length S of the drop shape results in the following set of three first-order differential equations with three boundary values, which is solvable by numerical procedures. The numerical fit of the theoretical drop shape to the shape recorded by the camera eventually yields the surface tension. The numerical fitting is a complicated process, but pendant drop tensiometer installed with DROP Image Advanced software offers a simple and elegant solution to determining surface and interfacial tension. Now, let's get familiar with the different part of the experimental setup. The schematic figure is shown as follows. The pendant drop tensiometer is composed of an IFT cell, consists of a light source, a chamber to load the two immiscible liquids and a camera to capture the high-quality images used for later analysis, a pump to inject the liquids, a controlling panel, two accumulators to store the two different liquids, and a data acquisition and analysis system. To conduct a new experiment, firstly, we need to do is to clean the IFT chamber and the attachments sequentially and repeatedly with hot water, 2-propanol and toluene, and flush the accessories with reverse osmosis water. Secondly, load the aqueous phase distilled water and hydrocarbon phase Bakken crude oil to two separate accumulators. Thirdly, do the calibration. By calibration, the program measures a known object, and the size of the pixel in the vertical and horizontal direction is calculated automatically. The calibration must be performed every time the magnification of the optical system is changed. Measuring the diameter of a cylinder placed in the camera's view usually performs calibration. A cylinder, a steel or glass rod or a tube may be used. There are three different ways to do the calibration: horizontal, vertical and sphere. Herein, horizontal calibration is the most commonly used one, which means that the horizontal width of a vertical cylinder is measured. The diameter is the size of the object and the value is taken from the initiation file. Another value may be entered, and a number of runs may be selected for an even better accuracy. When the OK button is clicked, the number of runs is measured and an average value from all the runs is used for calculation of the pixel size. In a horizontal calculation, the horizontal pixel size is measured and the vertical pixel size is calculated from this value and the aspect ratio. Fourthly, inject the aqueous solution into the chamber using the constant flux pump and check the aqueous level to ensure the later injected hydrocarbon droplet can be fully immersed. Fifthly, inject the hydrocarbon phase. During the process, a droplet will form at the tip of the needle. The volume of the droplet should be small enough to remain on the tip of the needle throughout the experiment, but large enough to provide a large profile and an accurate value of interfacial tension. The next step is to run the file and new experiment wizard in order to get the interfacial tension data. Export the report, close the software and clean the experimental area after the experiment. The easiest way to set up a new experiment is to use the new experiment wizard. A new experiment may also be started directly from the wizard. In the screen you select the type of experiment, surface tension sessile for our experiment. Pressing the next button takes you to the name screen. The name screen should be self-explanatory, both the parameter and method files, together with all the data files will use this name. If you select a name that already exists, the wizard will ask if you want to overwrite this experiment, which means you will not be able to use that file as a matrix for your new experiment. If you don't want to overwrite the files, you must either use another name or press the cancel button. The next button leads to the phase data screen where you select the name of the phases. The corresponding densities will be taken from the liquid's txt file. The next button takes you to the timing screen, where you set the number of measurements and the values for the experiment's timing. If you want to use a time file, pressing the Edit button will start the time file editor. If you save the data in that editor under a new file name, the new name will appear in the wizard's file name box. When you are satisfied with your choices, press the Finish button. The following information message is displayed. When pressing the OK button, you will be presented with the option to run the experiment right away. If you select Yes, the experiment will be started in the ordinary way. After setting the cursor position, follow the density difference. Click Measure to start the measurement. And the data will be shown in Results window. The details of each column are: measurement number, time from start seconds, the surface or interfacial tension, the shape factor, the radius of curvature at the drop's apex, the drop surface area, the drop volume, the contact angle at the drop limit, horizontal hairline. The total measured height from hairline to apex, the width, the number of optimization performed and errors or other messages, respectively. As for the task 1, students are required to determine the critical micelle concentration of the surfactant based on the given data. Generally, surface tension reduces with increasing surfactant concentration and then reached a plateau. Plot a figure with y-axis of surface tension and x-axis logarithmic axis of surfactant concentration. The intersection between the regression straight lines of the linearly dependent region and the straight line passing through the plateau is regarded as the critical micelle concentration. As for the task 2, students are asked to describe the changing trend of interfacial tension versus surfactant concentration, for example, in which surfactant concentration range, the interfacial tension decreases very fast and then reached a plateau at what concentration.